Good morning, dear students. I hope you all are fit and healthy. So dear students, now we are going to observe the anatomy of monocot stem. So we use the maize plant to observe the anatomy of monocot stem since maize is a monocot plant. So first of all, we will observe the transverse section of monocot stem through diagrammatic representation, and a thin transverse section of this monocot stem reveals the following structures. So outer one layer, that is known as epidermis, it is covered with the layer that is known as cuticle, same as we have observed in the dicot stem. But here the difference lies is that in the epidermis of monocot stem trichomes are absent. However, in dicot stem there was trichomes present. Then next to epidermis is a hypodermis. So epidermis, after that there is hypodermis. You need to remember that the hypodermis, in this the cells are sclerenchymatous. In comparison to dicot stem, there the hypodermis consists of collenchymatous cells. So hypodermis in monocot stem consists of sclerenchymatous cells. This is the difference between dicot and monocot stem. Then next is the ground tissue. So here directly after the hypodermis there is a ground tissue. In the ground tissue you are observing here is a parenchymatous cells. So ground tissue, it is made up of mass of thin walled parenchymatous cells will be present. And if you remember in the dicot stem you have observed that the ground tissue, it was further differentiated into cortex, endodermis, pericycle, but here in monocot stem ground tissue is not at all differentiated. And you are observing here the most important feature of the monocot stem is that the vascular bundles, they are scattered. So vascular bundles, they are scattered. This is the main feature of the transverse section of monocot stem. And the vascular bundles which are at the periphery, they are small as compared to the vascular bundles which are present towards the center. So this is the major feature of the vascular bundles. Next, if we consider vascular bundles again, the vascular bundles, it is surrounded by, you are observing here, thick layer. And this thick layer consists of cells of sclerenchyma. So this thick layer of sclerenchymatous cells is also known as bundle sheath. So this means bundle sheath is present in monocot stem, but bundle sheath was not observed in dicot stem. So again there lies a difference between the monocot and dicot stem. Next one is, in here vascular bundles, they are collateral, conjoint and closed. So collateral vascular bundles are observed. Collateral means phloem, it will be towards the epidermis and xylem will be towards inside. And other thing you should remember here in vascular bundle is that there is a conjoint arrangement of the vascular bundles, means both xylem and phloem, they are present on the same radius, they are joined. Then after that the vascular bundles are also closed. In dicot stem we have observed that the vascular bundles, they were open vascular bundles, but here in the monocot stem you are observing vascular bundles will be closed. Closed means there will be no cambium present between phloem and xylem. So if no cambium is present between phloem and xylem, then this condition is known as closed vascular bundle. And you know that this cambium, if present, it is responsible for secondary growth. Therefore, since here no secondary growth will be observed in monocot plant, since cambium is not present between phloem and xylem. So this is about the major important things you need to remember in the transverse section of monocot stem. So let us revise again. Here in the monocot stem, you need to remember that the epidermis, which is the outermost layer, it is not consisting, giving rise to trichomes, means trichomes are absent. However, if we consider the dicot stem, their trichomes was present. Then this epidermis, it was covered with the cuticle. And next to epidermis is a layer that is known as hypodermis. Hypodermis consists of cells which are mainly collenchymatous, but here the hypodermis, it consists of sclerenchymatous dead cells. Then after that, you need to remember the arrangement of the vascular bundles. That is, vascular bundles are arranged in collateral, conjoint and closed position. Closed vascular bundles means there will be no cambium observed between phloem and xylem. Then the most important thing, exactly next to hypodermis there starts the ground tissue. So this indicates that ground tissue is not at all differentiated into pericycle, endodermis and cortex as we have observed in the dicot stem. So this is another main important distinguishing character of the monocot stem. Next, if we consider the vascular bundles, they are in scattered position. So vascular bundles are scattered. This is also the very important contrasting feature of the monocot stem, and here you should always remember that the vascular bundles are collateral, conjoint and closed, as well as they are in the scattered position. So this is about the transverse section of monocot stem we have observed through diagrammatic representation. So now let us again observe the transverse section of the monocot stem using the picture. So in this picture, you are observing that the outermost layer is the epidermis, and this epidermis consists of barrel shaped cells, and this epidermis, it is further covered with the cuticle, with a layer that is known as cuticle, and there is no trichomes present. So trichomes are absent in epidermis of monocot stem. Then after that, there is a hypodermis just below the epidermis. This hypodermis consists of sclerenchymatous cells. So in contrast to dicot stem, the hypodermis was made up of collenchymatous cells, but here in hypodermis of monocot stem, the cells are sclerenchymatous dead cells. Then after that, you need to remember ground tissue is present, which consists of thin walled parenchymatous cells, and this ground tissue is not at all differentiated. As you can observe here very clearly, the ground tissue is not at all differentiated into different tissues such as cortex, endodermis and pericycle as we have observed in the dicot stem. So this is the another important contrasting feature of the transverse section of the monocot stem. Then after that, you should remember that the cells of the ground tissue, they have intercellular spaces as you can observe here. Then next to ground tissue, if we consider here vascular bundles, first thing is you are observing the vascular bundles, they are scattered, and the vascular bundles, they are collateral, conjoint and closed. First of all, we will consider collateral. Collateral means phloem, it is present outside, and this xylem, it is present towards inside. Phloem is present towards outside in the direction of epidermis, and xylem is present towards inside. Then conjoint means phloem and xylem, they are present on the same radius. This is the meaning of the conjoint, means xylem and phloem, they are joint. Then after that, there is another term, that is closed vascular bundle, means there will be no cambium present between phloem and xylem. Since no cambium is present between phloem and xylem, there will be no secondary growth observed in the monocot plant. Then another important feature of the vascular bundle is also that the xylem, it is arranged in the form of Y. So to understand the Y-shape arrangement of the xylem, you need to understand that there is a two upper rounded vessels that is known as metaxylem, and there was a inner xylem that is known as protozylum. So this condition is known as when protozylum is present towards the inside and the metaxylem is towards upper side, then it is known as Andark condition. Then next to protozylum, here you are observing lysogenous cavity. This cavity, lysogenous cavity, is basically formed by the disorganization of protozylum. So when one or more protoxylems, they merge or they are lost, then this cavity is formed, and this cavity stores water. So these are the major distinguishing features and characters of the monocot stem. So this is all for today. Take care everyone.